It is my pleasure to introduce our moderator, Dr. Amit Oza. Dr. Oza is the head of the Division of Medical Oncology and Hematology, a medical director of the Cancer Clinical Research Unit at the Princess Margaret Cancer Center. He's also co-director of the Drug Development Program at Princess Margaret, scientist at the Ontario Cancer Institute, and professor of medicine at the University of Toronto. Dr. Oza has been PI and co-investigator in over 100 clinical trials for gynecological cancer and advanced colorectal malignancies. He's also the past co-chair of the National Cancer Institute Gynecologic Cancer Steering Committee and executive member of the International Gynecologic Cancer Intergroup. Under his direction, the group has participated or led seminal studies in gynecologic cancer that have led to the approval or use of targeted agents such as PARP inhibitors and anti-angiogenic agents. Dr. Oza, thank you for joining us, and I'll turn it over to you now.
Thank you so much, Jay, and thank you for giving us the opportunity to brainstorm with you this afternoon. I'm going to give you a few sort of introductory slides just in terms of our translational research initiative that we sort of launched a few years ago, but just give some context. And you'll hear about ovarian cancer later on this afternoon. And we're going to be focusing on particularly around high-grade serous ovarian cancer, a really, really deadly disease, which unfortunately has a very, very high mortality rate. And the treatment options for this cancer remain somewhat challenging, and patients unfortunately present very late with disease. And despite lots of improvements in treatment, their disease is eventually the cause of their premature deaths. And unfortunately, about 330,000 women annually succumb to ovarian cancer on a worldwide basis. So a really aggressive disease where we need to make better strides in terms of treatment. A few years ago, we had the opportunity to put in for a translational research initiative through the OICR, and we thought that it would be a good idea to reflect on what did we actually achieve through that TRI, and what are some of the sort of key sort of messages. So we sort of really looked at our ovarian TRI as a way of looking at therapeutic vulnerabilities in high-grade serous ovarian cancer, and had three different components to the disease, high-grade serous ovarian cancer. We were also looking at biosynthetic stress pathways in terms of leveraging vulnerabilities, and also increasing immunogenicity in high-grade serous cancer. And you'll hear about some of the different clinical trials that were embedded into this triad of questions that we had developed. We also underpinned the translational research initiative with a longitudinal assessment in terms of patients who develop disease and resistance over a period of time, and a platform trial called BioDiva, which went through the length of time that patients undergo disease and treatment and are assessed in terms of changing patterns of resistance, and then that's the aim, to collect tissue over a long period of time. The translational research initiative was a sort of really a deep assessment in terms of what could we actually learn about both ovarian biology and use that information in terms of directly intersecting with the disease in terms of treatment approaches. And many of the folks are on the call, and you'll see some of the work that has been coming through this translational research initiative. This was a very multidisciplinary approach, looking at really basic translational research, deep assessment of the biology of disease, and really trying to intersect it directly with bench-to-bedside approaches and back again in terms of understanding the changing biology and how we can actually leverage that into therapeutic opportunities. And you'll hear about both of those later on this afternoon. The translational research initiative, which was funded by OICR, was hugely successful and led to really very, very major sort of collaborations over the course of several years. This led to more than 15 major collaborations with institutions and industry partners, including major sort of partnerships with Providence, which is an RNA therapeutics company. During the course of the study, we have sort of garnered specimens in over 1,800 specimens, which have been collected in multiple different clinical trials, which are being analyzed. The study is the TRI was also leveraged with lots of additional sources of funding and grants, with about $25 million of additional grant funding concepts that were submitted for funding, and has led to 25 publications. There have also been multiple different clinical trials and translational research studies that have been launched through the course of the study, and over 740 patients have been enrolled in multiple different studies over the course of the TRI. And there have been several different awards that have been awarded, and I think we've counted about 20 research awards that have been garnered by the team. Lots of samples collected from all of the studies, and it's also important to recognize that the dream team that we had sort of gathered for this have actually been recipients of 4 Canada Research Chairs, as well as a chair in ovarian cancer research by Stephanie. It's also been fantastic opportunities in terms of training the next generation with 6 graduate students, 21 doctoral students, and 38 fellows that have actually participated in different components of the TRI team research over the course of the last 5 years. And there have been sort of major initiatives that are leading to commercialization. Repair was a sort of major initiative that came from the work of Dan DeRocher in terms of ovarian cancer resistance in high-grade serous disease. And you'll hear later on about GCN2, which is being led by Rob Rottapel. So fantastic collaborations that have led to amazing achievements over the course of the last 5 years. And this slide very briefly sort of touches on the sort of key areas where we've been sort of working in terms of overcoming resistance and major milestones. And you'll hear about some of the sort of key milestones later on, but really lots of different developments coming out of the specific projects, and Stephanie will touch on some of the key ones from the clinical trials, and Rob is going to have further discussion about GCN2 later on. The one other thing that has come from the TRI has been sort of, again, fantastic collaborations and training opportunities for many of our students as well as fellows, and these have spanned multiple different continents, as well as lots of collaborations in terms of clinical trials that are going on between Canada, US, Europe, and Australia and New Zealand. Have a platform study which really looks at both longitudinal assessment in patients, but also multiple sampling time points, both opportunistic as well as planned, looking at collecting blood and tissue and ascites, particularly for biomarker development now and in the future. So there's a significant treasure trove of specimens matched with clinical trial data and outcomes that we can actually do deeper mining in over the course of the next few years, which we've garnered as part of this TRI initiative. So just in terms of looking towards the future, I think we're sort of looking at further development work with GCN2, which you will hear about. There's a lot of work currently going on in clinical trials through some of the work that started with Dan DeRocher, and this has led to development of therapeutic products through Repair, which has become a major force in terms of looking at overcoming drug resistance in high-grade serous ovarian cancer and looking at leveraging additional therapeutic vulnerabilities. Definitely will also touch on Revolve, which is actually trying to push further how we overcome PARP resistance in ovarian cancer. and she'll talk about PHL93, which is again trying to overcome resistant and refractory high-grade serous ovarian cancer using WEE1 inhibitor in conjunction with chemotherapy. So a lot of work that needs to be done, but I think that the key area where we sort of really want to sort of focus on is how we can actually link preclinical work with clinical work and biomarker development, optimize real-time patient decisions with real-time genomics and immunogenic information, which is what we're trying to do, and really highlight the importance of teamwork in this collaborative framework, which actually will allow us to push forward in terms of improving outcomes. We're not going to take questions now. I'm going to hand it over to my colleagues who are going to sort of go through the next steps. I do have a confession to make at the moment. I'm in the middle of my clinic and I have to go and see a patient with ovarian cancer. So I'm going to take your leave for about 20 minutes whilst Stephanie is giving her talk. I have gone through her presentation, so I'm aware of what she's going to talk about, but I will come back in about 20 minutes for the next part of the discussion.
Thank you. So it's really my pleasure actually to follow up and actually present some of the clinical translation that we were able to do through the TRI ovarian cancer. And I really want to take this opportunity to thank OICR for the support. That was actually tremendous, and we have come quite along, and we have made a significant progress for our patients. So a deep thank you. So as I was presented, actually the TRI is really a multidisciplinary team, and really have three big focus on the project one, which was really the genomic instability that I led with Dan Rocher. The project 2 that was led by Rottapel with the biosynthetic stress, and the project 3 mainly in the immunogenicity. So I will go about all these different area and see what we have done in each of these in terms of clinical translation and how that we can impact our patient and what did we learn. So the first one is definitely the DNA repair. And actually in the DNA repair, like high-grade serous ovarian cancer is a perfect model for the DNA repair. We use a lot of platinum-based chemotherapy that induce some double-stranded DNA. And then we know that we need the homologous repair pathway to repair the double strand. But if we also actually inhibit the PARP inhibitor treatment, you really have this concept of synthetic lethality. You actually focus on these two DNA repair pathways that actually lead to the cell death. And specifically in high-grade serous ovarian cancer, in the homologous repair pathway, BRCA1 and BRCA2 play a tremendous role and actually lead to all the development of PARP inhibitor treatment with a significant benefit for the patient with a high-grade serous ovarian cancer and BRCA mutation. So we really, based on that, we actually, through the TRI, we were able actually to try to learn more. And we have seen in clinics that some patients who have a BRCA mutation but do not respond. And in the contrary, they do not have a BRCA mutation but respond really well. So actually, through the TRI, we actually build this specific cohort of patients to understand the extreme responder to olaparib. And we have built that multi-center through Canada, but also internationally. And we have Spain, UK participating, Australia in this specific collection, looking at the extreme responder of olaparib, which was defined as patient responding more than 18 months, and the patient that did not respond, which are less than 6 months. And actually, this is now completed. We have 50 patients in each cohort, and we are ready actually to analyze. And as you can see here, the patient who have this long-term benefit definitely have a long overall survival, contrary to the one who actually have this poor responder to PARP inhibitor and really have a short overall survival. So it definitely something that we are going now to the next step and do deep characterization of the tissue and blood that was collecting on this cohort. The next trial, actually, we were able to launch through this TRI opportunity is a specific window of opportunity for patients who have a high-grade serous ovarian cancer, platinum-sensitive recurrence. It seems to have potential secondary debulking surgery again. So for this patient, they were not selecting on specific biomarker, just the fact that they have platinum-sensitive recurrence, and we think we can debulk them again. For this patient, they had a baseline biopsy and the blood collection for ctDNA. And then they received olaparib for 6 to 8 weeks and then went to the debulking surgery. At that time, we collect again the tissue and also the ctDNA collection. After the surgery, the patient were enrolled in the platinum-based chemotherapy followed by olaparib, or no chemotherapy at all and just olaparib alone. And again, during this time, blood samples were collected at different time points, and there is a mandatory biopsy and ctDNA collection at the time of progression. The trial will be very informative in terms of understanding the disease biology. We have now 44 patients that were enrolled. All the tissue has been collected. We have fresh frozen tissue. We have FFP collected. And really, we want to look at the early proliferation adaptive response to olaparib and see if we can identify some specific biomarker of therapeutic response. So that is a really rich trial that we now looking to assess very deeply in terms of potential pharmacodynamic biomarker. The next trial actually that we conducted as part of the TRI as well was the EVOLVE study, which was a very different context. It was patients here that initially were treated with a PARP inhibitor treatment and they progressed. At the time of progression, they had a baseline biopsy, and then these patients were treated with the combination of cediranib and another PARP inhibitor, olaparib. So the idea was really to see if patients were progressing on the PARP inhibitor, can we re-challenge with PARP inhibitor in combination with an anti-angiogenic. And for each patient, we have the baseline biopsy at the time of PARP progression, and we also have the tissue at the initial diagnosis, which was treatment naive. As part of this trial, we enrolled patients with platinum-sensitive disease, patients who were called platinum-resistant, and patients who actually got prior PARP and then got another type of treatment. So what did we learn? We learned quite a lot, actually, from this clinical trial. We actually were able to assess the tissue and the clinical outcome. And this paper was actually already published in Clinical Cancer Research. And as part of the trial, we also have ctDNA collected as part of the trial. And this longitudinal ctDNA collection, which was done in collaboration with the whole exome sequencing of the tumor, we have currently submitted the paper to Clinical Cancer Research. So what did we learn from this trial? Some patients actually had benefits of this re-challenge and some did not. And we tried to understand from the analysis of the baseline biopsy the change that was observed in this tumor tissue, because definitely high-grade serous ovarian cancer is still defined as a high-grade, but the mechanism evolved. And what we have seen is that patient actually can have the reversion in the HR gene. So a patient that initially was called to be BRCA1 mutation can actually reverse and not be longer homologous repair deficient, but be homologous repair proficient and begin to be PARP resistant. We also see at the time of PARP resistance some cyclin amplification. And again, we can debate of the definition of cyclin amplification in terms of the number of copy. We also have seen at the time of PARP progression that we have this upregulation of the multidrug efflux pump gene ABCB1. And what is interesting is actually the patient who have at the time of PARP progression this reversion mutation in the HR gene, which was in BRCA1 and 2, who actually also in BRCA51, definitely had a worse outcome. They did not respond to any other treatment. And also what is interesting is that this mechanism of resistance actually can be seen in multiple reversion can be seen in one patient. So definitely the disease evolving, and we need to actually adapt to that and try to see if we can identify this kind of mechanism of resistance in a timely fashion. So if we want to summarize, and actually based on all this trial we have done, we were able actually to learn more about how to overcome resistance and see after what is the next trial we need to do for our patient. And actually we have published 2 reviews based on this previous work, and we have actually identified 4 big mechanisms of resistance to PARP inhibitor, which was the restoration of the HR pathway, the replication fork protection, the signal transduction pathways, is other pathway involved, and also the upregulation of the drug efflux. And as part of the TRI, we definitely try to look at these different aspects. So as part of the TRI, I present to you the EVOLVE study that was already published. And we have this NEO trial, which was a window of opportunities that we really look specifically at this restoration of the homologous repair. We also have this extreme responder, which we will look at the other pathway involved in the extreme response. And now what I want to focus is actually in collaboration with Dan DeRoche, we were able actually to have specific analysis of this replication fork protection and really have specific investigator-initiated trial. And why these are very important, because initially when we look initially at my schema, I really say that a high-grade serous ovarian cancer what is important is a DNA repair pathway, and you have different type of pathway. But what is also very important is this DNA repair pathway is definitely important and in link with the cell cycle. The cell cycle is really a big component of the DNA repair pathway and play a significant role. And why it's so important is because it has this TP53 mutation, and because of this specific vulnerability, you already have a dysregulation of your cell cycle. In high-grade serous ovarian cancer, you have this TP53 mutation, so you lost this fidelity of the G1S checkpoint. So this cell really depend more on the G2M checkpoint of the cell cycle. And that, if we look at the GM2 checkpoint, you definitely have multiple kinases that you can target to really try to dysregulate the cell cycle, which will lead to the synthetic lethality. And if we want to focus a little bit more, we can look at the specific ATR-CHEK1 pathway as a potential therapeutic target in high-grade serous ovarian cancer. Because if we induce some DNA damage, yes, you activate the DNA pathway, but you really integrate your DNA signaling and you activate your cell cycle. And here, one of the kinases that we will focus on first is the ATR. And there's several ATR inhibitor treatments that are currently in development, but I want to highlight one. As Dr. Oza mentioned, through the work with Dan Roche, we were able to collaborate with the Repair Therapeutics, and actually we were able to actually conduct the phase 1/2 trial with this specific RP3500, which is an ATR inhibitor. The phase 1 was conducted here at Princess Margaret in a specific disease where the DNA damage repair loss of function mutation was identified. And this was given as single agent. So this ATR inhibitor was given as a single agent in this specific tumor who have this repair loss function mutation, such as ATM, BRCA1, BRCA2. And as you can see here, as a single agent, we have seen some signal of activity. And if we take specifically the ovarian cancer patient, it was 20 patients in this early phase, the response rate was 25%, with a clinical benefit rate of 75%. And what is very interesting, actually, is that these patients were platinum resistant and were post-PARP inhibitor treatment for the majority of them. So definitely targeting this pathway may actually overcome the platinum resistant and the PARP inhibitor resistance. And the response was observed in a patient who has this specific loss of function of alteration in BRCA1, RAF51C. The question was, can we see this kind of response targeting this pathway beyond the specific biomarker of loss of function tumor? And actually, that was actually assessed in this specific trial where I didn't target the ATR, but we target the WEE1. And here's the same rationale, is really trying to target the cell cycle is dysregulation. We have a high-grade serous ovarian cancer that really rely on this specific G2M phase, and then you can actually target the ATR we saw before, but we can target the WEE1. So if you target this specific checkpoint, you have this specific checkpoint with a TP53, and then we can increase the replication stress by adding gemcitabine, which is another really target all the checkpoint of the cell cycle that will lead to the mitotic catastrophe. So it's what we wanted to assess a specific hypothesis. And actually, a clinical trial was conducted in platinum-resistant high-grade serous ovarian cancer with no prior line of treatment. And again, the same principle as all the previous investigator-initiated trial we did is actually to have baseline biopsy that we can compare to the archival tissue to see if there's any specific change or specific biomarker that we can identify. So the trial was conducted assessing the combination of gemcitabine plus the WEE1 inhibitor versus gemcitabine plus placebo. And that actually validates the concept of this synthetic lethality and the interest of targeting the cell cycle deregulation in high-grade serous ovarian cancer. Because when you use gemcitabine alone in a patient with high-grade serous ovarian cancer, the response is only 6%. And when you combine with the specific WEE1, the response is increased to 23%. And what is interesting here, as you can see here in the little star, it's even effective in a patient called platinum refractory, which are really a niche of patients that did not respond to any current treatment as a standard of care. And also what you can see here is that this benefit can be prolonged. So again, we went back to the tissue that was collected and what are the potential biomarker of response. And as you can see here, the patient who had the best benefit of the combination of the WEE1 plus gemcitabine were actually the patient who had the cyclin E amplification. So where we are currently, we're designing the next step of the clinical trial. And as Dr. Azad quickly mentioned, we have designed this REVOLVE study, which we want to assess in a real time, what are the mechanisms of resistance for patients? And do we have a cyclin amplification? Because the cyclin amplification seems to mean something, and maybe a specific biomarker. And from the EVOLVE, we saw that who have a BRCA reversion is really have a poor prognosis. So the idea here, the trial is currently ongoing. We already have 3 patients on, is looking at patients with a high-grade serous ovarian cancer who progress on PARP inhibitor treatment. As you saw in EVOLVE, the platinum-resistant to platinum-sensitive didn't really matter in terms of the mechanism of resistance. So here it will be platinum agnostic. And then the patient will have a tumor biopsy and the ctDNA collection. While we wait for the result of this whole genome sequencing on the tumor and the ctDNA, the patient will receive treatment with niraparib and bevacizumab, which is similar to the olaparib and cediranib combination, is trying to re-challenge with a PARP combined with an anti-angiogenic. And then we have the result back of this whole genome sequencing. And here we'll adapt the treatment. If we cannot see any alteration, then the patient will continue on the PARP with the anti-angiogenic, but we will add a PD-L1 inhibitor to try to really increase the response and really target the microenvironment. If actually we have identified some version, then we will stop the PARP inhibitor treatment because we know now that will not be effective, and we'll replace that by weekly paclitaxel chemotherapy that recently has a hypothesis show that it can be beneficial in the BRCA reversion in combination with an anti-angiogenic and an immune therapy. So that is really a novel way of doing a clinical trial. And again, I want to thank OICR for the support. And definitely, as we can see here, cyclin amplification is definitely a good target. And working back on the lab and with Dan Roche, as we can see here, again, if we go back as a cell cycle theory, if you have a cyclin amplification and a TP53 mutation, you really have an abnormal cell cycle, which increases the replication stress and the reliance on the other cell checkpoint. So here, imagine if you have a cyclin over amplification or overexpression, you can imagine, again, we discussed the WEE1, we discussed the HER, but there's another kinase, which is a PKMYT1, that may be a potential target in this specific context. And Dan Roche work with a large-scale CRISPR screen actually identify that the cyclin amplification is synthetic lethal with a PKMYT1 kinase inhibition. So this work was published in Nature last year, and this finding actually led to the design of a specific molecule targeting the PKMYT1 kinase inhibition, which is called RP6306, again, in collaboration with the Repair Therapeutics, and the phase 1 is currently ongoing. So definitely a lot of has been achieved through the TRI ovarian cancer and definitely impacting our patient. And I think it's just the beginning. Now we will move on from the project 2. And again, Rob Rottapel is on the line, and then we'll further discuss about the project 2 and where it's landing. So I just want to highlight this specific project on the biosynthetic stress that we have actually conducted from the clinical finding to the clinical trial as well. And again, in vitro, his lab has shown that atroconazole and chloroquine can actually have a synergistic activity and be a potent anti-tumor activity by affecting the lysosomal function. Based on this finding, actually, we conducted a phase 1 combination of these 2 drugs. And again, we combine that with mandatory baseline biopsy and on-treatment biopsy to identify specific biomarker. Unfortunately, we didn't see a signal of activity, but doing the pharmacodynamic marker on the paired biopsy as part of the trial, we understand why this was not seen as effective is because in the tumor, we didn't see a pharmacodynamic impact. So the drug were not able to reach the tumor. So really show the importance when we do this clinical translation to build as part of your protocol biopsy and blood sample to really assess the activity. I will not go too much detail. The work has been published and is in open access. So if you are interested, please do not hesitate to reach out to this article. On the next project was actually the project 3, which was really trying to target the macroenvironment, specifically in the immune space. And currently in ovarian cancer, and specifically in high-grade serous ovarian cancer, unfortunately, we didn't see the activity of the PD-L1 inhibitor that was seen in other disease type. Unfortunately, currently, to date, all the trial that was conducted with PD-L1 as single agent didn't show a significant benefit in our patients. So definitely, the high-grade serous ovarian cancer have the cold microenvironment, very immune suppressive microenvironment in high-grade serous ovarian cancer. So actually, as part of the TRI, we also designed a specific study trying to see if we can overcome this resistance and actually increase the potential of immunogenicity in high-grade serous ovarian cancer. And here the idea was to try to combine the PD-L1, which is pembrolizumab, with a specific vaccine, which targeting the survivin. And we know that survivin protein is overexpressed in high-grade serous ovarian cancer. So we were actually trying to increase the immunogenicity by combining the pembrolizumab with the specific survivin vaccine and this oral cyclophosphamide metronomic. The phase 1 dose escalation was initially conducted in platinum-resistant ovarian cancer, and then specifically in the platinum-resistant high-grade serous ovarian cancer. And again, as all the ITT trials that we have conducted through the TRI, biopsy was mandatory and also blood collection. And here specifically, given the immune, we build a lot of PBMC. So what did we observe? And I want to highlight that actually this work has been presented at ASCO last year by one of our fellow, Anna. And actually, what did we show? We show that some actually high-grade serous ovarian cancer can have benefit of this treatment, and the treatment actually can persist with time. And again, it's very difficult here to see which benefit or not of this potential combination. And it's true, there's some patients that are not responding. So again, very important to go back to all the criteria to try to distinguish who responding, who are not responding. And actually, we learned quite a lot. So it's still a work in progress. I just presented a portion of what is currently ongoing, and that is just pilot data. But when we look at the specific PBMC, actually what we see is that actually we can have some stimulation that can be seen on the PBMC. And we can see actually for here you have a patient, we have one patient who have a complete response. We have a patient who have a partial response. Then we have some patient with stable disease and one that did not respond at the interferon gamma level in the PBMC. And what you can see here is definitely for the patient who has this complete response, you have an immune response specifically against survivin. So definitely there's a possibility that actually we can reactivate the immune system in ovarian cancer. And when you have this complete response, actually the response is very high and long lasting against survivin. But in the other patient, we definitely have low or negative immune response. Again, a lot of work is still ongoing. The patient were not screened for the LHA matching, so maybe it's something that we need to consider, but a lot of work still in progress, and we will be happy to follow up on this work when we have more data. But definitely a lot of interest, and really show that despite the immune suppression agent, we can definitely have some response in ovarian cancer, so quite encouraging. And again, I want to also, after finish by the specific slide on BioDiva that Dr. Oza just mentioned at the beginning. Really, our program really try to understand the disease for every patient. So every patient that come in clinic, we want to understand why this patient will respond or why she will not respond to this specific treatment. So it's how BioDiva was built, and actually we collect tissue, we collect blood at the different time point of the patient journey. And currently, we have 441 patients are currently enrolled in this specific program. And you can imagine if we go back after to this specific biobank that is clinically annotated, we can learn a lot about potential biomarker, and it's a really discovery platform. Currently, we have samples collected over time. So we have 6,000 blood collected, 900 tumor, and also collected fluid. And also, what is also important, we actually try, and thanks to the TRI as well, well, we were able actually to create some preclinical model for high-grade serous ovarian cancer, and that is also work that we have done in collaboration with Dr. Rottapel to really try to see if we can actually have some patient-derived PDX model to then learn more and look at specific mechanism of resistance to response and have some specific drug screen. So we currently have 25 PDX model, and we also try to build on the organoid model, and maybe we can discuss that after, and Dr. Rottapel can give more information about this specific preclinical model. But definitely, I want to finish by really thanking OICR for the support, and really, I hope I give you a sense of how much we come thanks to this initiative that was started several years ago. And I want to thank also our patient and clinical and research team. Thank you.
Thank you very much, Dr. Lheureux. Our next speaker is Dr. Kyle Francis. He will represent. He's currently a research associate in the laboratory of Dr. Rob Rottapel at Princess Margaret. He received a doctoral degree from University of Edinburgh, Scotland, under the supervision of Dr. Simon Langdon, where he studied checkpoint kinase 1 inhibition in ovarian cancer. He's currently studying the kinase SMG1 in ovarian cancer and developing new SMG1 inhibitors for disease treatment. Kyle, thank you for joining us. I'll turn it over to you.
All right, so I'm a research associate in the lab of Dr. Robert Rottapel. So I'm studying nonsense-mediated decay as an adaptive stress pathway in high-grade serous ovarian cancer. So just a quick review. I don't have to go through much of this because Stephanie and Amit mentioned it earlier. So I'm studying high-grade serous ovarian cancer, which is characterized by p53 mutations and a lack of homologous recombination DNA repair, such as roughly like 50% of the tumors when it was analyzed in the TCGA. Just gonna get my pointer out. Yeah, so besides these lack of HR repair, the tumor often gets widespread genomic copy alterations. So you can see this on this Manhattan plot from chromosome 1 to 23. Besides that, there aren't a lot of targetable oncogene drivers. Therefore, we set out to do these genomic screens on a panel of roughly 50 high-grade serous ovarian cancer cell lines. So before I get into that screening data, I'm just going to go through a little bit about the disease. So the proposed cell of origin is the fallopian tube cell epithelium within the ovary. And what happens when they transform into high-grade serous ovarian cancer, you get the acquisition of these fitness properties that promote survival, proliferation, motility. Those are often seen in the hallmark of cancers. However, with these fitness properties, they also get a byproduct of stress ranging from everywhere from the genome to the proteome. And it's from there that we think that these high-grade serous ovarian cancers tumor cells are adapting pathway to alleviate the stress. So that we think if we could identify these adaptation pathways, we could find new vulnerabilities for treatment. Just going into the screens we generated. So it was 50 ovarian cancer cell lines. We did a whole genome with an shRNA library targeting roughly 16,000 genes. For those that drop out quick, we deemed them highly essential genes that are required for high-grade serous ovarian cancer tumor fitness. What we found in one of them was the SMG1 kinase was essential in roughly 75% of our panel. In vitro using secondary assays, but also in vivo, just to show that SMG1 is required for tumor growth. So what is SMG1? So SMG1 functions in this pathway called nonsense-mediated decay. Its role is to act as an mRNA quality control pathway that degrades these transcripts that contain additional premature termination codons. So what would happen if the cell had these in abundance? You would get translation of these truncated and often misfolded proteins into genotoxic stress. So just going quickly into what the pathway is. So when the ribosome surveys and identifies one of these PT sites, it'll stall and you'll get the interaction of the ribosome and UPF2 at this exon junction complex. You'll get the formation of this SIRF complex containing roughly around 15 proteins. Those of interest are SMG1 and UPF1. So the SMG1, which we identified in our dropout screens. So what SMG1 does here is it phosphorylates UPF1. UPF1 phosphorylate acts as an anchor for endonucleases and 5' decapping enzymes, leading to the destruction of these targets. So going into the molecular biology of the pathway. So this is just a quick schematic looking at the phosphorylation sites that SMG1 does on UPF1. NMD, those of importance are mainly the C-terminal targets. The one we're interested in that we found is tightly correlated with the activation of NMD is this serine 1116. So what we find is that when we look at these levels of this active NMD marker, phospho-UPF1, we found that it's highly activated in a large panel of high-grade serous ovarian cancer cell lines. However, the signal is fairly low in our normal FT model, suggesting that this pathway is hyperactivated in high-grade serous. So if we have highly active NMD, we want to be able to shut it off. So what we discovered is through OICR's chemical group, they actually had an SMG inhibitor from Wyeth Pfizer. It's called 11J. What this can do is, so this is a baseline level of the NMD marker in OVCAR3 cells, and that when you treat it with increasing amounts of 11J, you can actually shut off the NMD program and stabilize the targets such as ATF4. So this is a 6-hour treatment, so it's good that we have a tool for the SMG inhibition. If we look at the cells later at a time point of like 48 hours, we can see that it's inducing the cleavage of caspase-3. So you can see that right here for the 11J treatment relative to DMS. So suggesting that when we shut off NMD through an SMG inhibitor, it induces apoptosis. Now that we have a chemical compound, we want to see its sensitivity of cell lines. So I'm only showing 2 here, high-grade serous cell lines, but also we have some of those organoids that Stephanie mentioned from the Princess Margaret Living Biobank. We can see is a good sensitivity around like sub-micromolar amounts for the high-grade serous ovarian cancer models. However, the immortalized FT normal models are relatively insensitive. So this was interesting for us because it shows us that there's a therapeutic window for this new adaptive stress target. I'm not showing it here, but we have tested other normal models such as cardiomyocytes and normal pancreatic cells, and they show relatively the same trend as the FTs, highlighting again our important finding. So that leads to the hypothesis that the damaged genome in high-grade serous ovarian cancer that's characteristic of the disease will lead to a damaged transcriptome and the activation and dependency of NMD. However, if we inhibit this pathway, we should be able to stabilize these damaged transcripts and those damaged proteins leading to cardiotoxic cell death. So now that we identified an essential pathway, we want to know which of those NMD targets are driving the cell death phenotype. So in order to do that, we did an RNA-seq approach, a 2-prong approach. So we did bulk RNA-seq for 11J treated and DMSO control cells. And we were able to find that when we inhibit this pathway, we get a stabilization of roughly 6,600 transcripts. And then to combine with that, we also want to know which of the transcripts are being actively degraded by the NMD pathway. So we took that phospho-UPF antibody, we IP'd the ribonucleoprotein complexes, and we eluted the RNA. And we sequenced that and did differential expression analysis compared to IgG control. Found roughly 4,000 are bound to the active complex. So when we take the union of that, we're able to identify roughly 2,000 NMD targets within this high-grade ovarian cancer model disease. So I'm just going to present some data, taking a deep dive on these targets just to see what's going on in these cells when we treat them. We did an overlap of a gene set of proapoptotic gene targets. We're able to find roughly 24 are being stabilized upon treatment with 11J. We could actually validate these with qPCR just to show that they are increasing in a dose-dependent manner within the OVCAR3 cell line, showing that stabilization of these transcripts is likely leading to apoptosis. And I just have this summarized right here. So we think that NMD is driving tumor progression by silencing these proapoptotic NMD targets. So we know what's being stabilized and what's being targeted by NMD, but we also want to realize this polysome RNA sequencing. We isolated the fraction containing more than 3 polysomes, so this would be our highly translated fraction for DMSO and 11J. And from there, we did a similar thing doing differential expression analysis just to see which of those are being highly translated. Before I get to that, just one thing we did with the NMD targets is that we looked further into their transcript biotypes in the RNA-seq. We find that a majority of the targets are protein coding, which is in agreement with the literature, but we also found a lot of these damaged transcripts that we call them. So like the ones that contain retained introns or splice variants inducing premature termination codons. We also have a class of non-coding RNAs, so like processed transcripts, long non-coding RNA, and pseudogenes that are also targets and stabilize when we inhibit the pathway. When we looked at the polysome RNA-seq, we would have expected only to see a majority of those protein coding being increased in translation. But surprisingly, we found a high percentage of the damaged and the non-coding RNA. So this is pretty interesting because our collaborator, Claude Perot, has identified non-coding peptides on the aminopeptidome, which I'll explain in the next following slides. Yeah, so here's the collaboration with Claude Perot. So we know which are the NMD targets are highly translated, but which of them are actually being presented as peptides or antigens on the MHC class 1 molecule. So what Claude does is he takes our SMV treated and DMSO control samples, CIPs, MHC class 1, elutes the peptide and identifies it by mass spec. We're able to find a total of 5,300 MHC class 1 antigens. Of those, roughly about 2.5% are increased in presentation. So these would be our NMD regulated peptides. So now that we have 133 of interest, these would be our candidates for CD8 T cell activation. We want to take some further into studies. So of those 133, first we want to see which of them are just expressed on the tumor. So this would be our tumor specific NMD regulated antigens. We use this database called Genotype Tissue Expression Database that they use in Claude Perot's lab to see which of them are only expressed in the OVCAR3 model. So those are all these right here. So they're not expressed at all in the periphery. What else here? So there's a combination of coding antigens and mutated and non-coding antigens. And just happenstance, a lot of them have B702. So we want to take 4 of these for follow-up. These are strongly responsive to NMD. And we want to see if we could do a proof of concept experiment to show that these are functional when you sort for antigen specific T cells. Before doing that, because I'm showing a lot of our data just for limited to OVCAR3 within this 50 minutes, I just want to briefly mention that a lot of these antigens, their transcripts are regulated by NMD and other models. So we don't think this is just one type of tumor cell type or cell line that's presenting these antigens. We're also looking into other models such as the pancreatic model, CFNMD also regulates these antigen transcripts. So this is encouraging for us. And we're following up with this with Etienne Caron and Montreal. So getting back to the proof of concept experiment, we want to examine the whole T cell repertoire. So we looked at HLA matched cord blood. We isolated the cells of interest, double checked that it's stained with HLA B72. And what we want to do here is just to see if we could pull down antigen specific T cells, but also show that the cells also induce killing of presenting cells presenting those antigens. The way we isolated the antigen-specific T cells, we use these dextramers that contain a fluorophore, our HLA of interest, and our antigen that I showed in that table before. And from there, we're able to sort a small population of antigen-positive cells and expand them in vitro to cell amounts that we're able to perform the T cell killing assays. This is done in collaboration with JC's lab at the Department of Immunology in Toronto. So just briefly going through the assay. So we're able to label our APC. So just because we want to see if the peptides themselves are functional, we're taking a antigen presenting cell that could be easily killed if presented with the right specific TCR. So we use these EBV transformed B cells. We're able to label them with CFC low for those that are unpulsed and CFSC high for those that are pulsed with our peptide. When we incubate with different ratios of our antigen specific CD8 T cell, we should be able to see a reduction in this high peptide pulse APC. So we tested about 4 of those T cell populations. Here's an example of one of them. At the higher ratios of T cell, we're able to see a good amount of killing of only those that are pulsed. So this was encouraging for us because not only can we show that our inhibitor treatment can present novel antigens, but their corresponding T cell in the human body. Here's an overall summary of the data. So you're able to show that the damaged transcriptome is translated using the polysome-seq. We're able to see cell death due to proapoptotic genes being NMD targets, but also to show that we could identify immunogenic tumor-specific antigens. This is just the Rottapel lab. Thank you.
Thank you very much, Carl. Our next speaker is Dr. Methvin Isaac. He's representing the therapeutic innovation themes. He's a senior scientific advisor in the Drug Discovery Program at the OICR. He has over 20 years of experience in identification of drug candidates across multiple therapeutic areas, CNS, GI, and oncology. As a medicinal chemist, he has made significant contribution to the discovery of several NCEs, including 6 drug candidates that were advanced into late-stage preclinical and clinical development. He's an inventor of over 75 issued and published patents, an author of over 35 research papers and review articles in peer-reviewed journals. Methvin, thank you for joining us. I'll turn it over to you.
Okay, so thanks, Jay, and everyone for inviting me. Again, continuing the theme of looking at therapies for ovarian cancer. I mean, one of the things I've learned, luckily, learning a lot from Rob and a lot of the insights that he has from, therapy. So it seems like a tumor like ovarian cancer appears to be very heterogeneous. And so, a lot of times when you go after certain specific cancer cell, and, it's just a matter of time before resistance come about. And so there's this emerging theme now where instead of kind of just targeting one, if you could actually target sort of the bulk microenvironment and just create, stress, then that might be a more effective way of targeting, a tumor like ovarian cancer, which is tough to treat. That basically going after nodes within 1 or 2 cells in the population. And so with that said, Rob, brought this target to us, GCN2, with the notion of really going after cancer cells and, targeting, the intrinsic stress, trying to avoid any sort of method that these cells might try to adapt in order to escape cell death. And so this target, as I mentioned, is called GCN2. Okay, and so the name GCN2 comes from general control non-irrepressible 2 kinase, or short form GCN2. And I wanted to, on this first slide, just give you a general idea of what the target is. So it's a kinase, it's a very large kinase. You could see almost 650 amino acid. And one of the interesting features of GCN2 is that when it gets activated, it actually forms this GCN2 dimer that I'm showing here on the right-hand side of the screen. So that dimer system is actually what's driving a lot of the catalytic function. And so just some key points to learn about GCN2 as it applies to cancer cells. Now, first, note that cancer cells tend to be addicted to what we call non-essential amino acid. And I just give an example here, asparagine. And so this is actually important because, as I say, one of the challenges a lot of times in therapy is to be able to establish a therapeutic window between the tumor cells and normal cells. And one good way of doing this is actually, if you know that tumor cells are really, really dependent on non-essential amino acids, whereas the normal cells wouldn't care, you could see there's an intrinsic built-in therapeutic window. If these tumor cells become highly addictive to the non-essential amino acid, and then you can use that as an Achilles heel. So GCN2 serves as a sort of a sensor for amino acid availability. And so by knowing that, it means then that GCN2 could indeed be a very, very interesting target to go after in therapy. To this audience, there's no need to express how much of a need exists for the treatment of ovarian cancer. It's one of the #1 killer for gynecological cancer. The market is, for it is really, really huge, and there's a huge demand for better therapy. I mean, you could see from a lot of the literature, with all of the promise of immunotherapy, immune checkpoints, a lot of those haven't actually worked as well in ovarian tumors. So we are really, really in desperate need of better. So if you think about then, ovarian cancer cells and the role that GCN2 might play in this type of tumor. Now, ovarian cancer cells, are exposed to a number of stresses. And so I just highlight a few here. There's reactive oxygen species, hypoxia, there's unfolded protein stress, but more important and going to be the focus of this conversation is the nutrient deprivation. They become highly, highly dependent on essential nutrients to really drive their growth. And so you could imagine that this stress biology could be a fundamental way to really going after treating some of the resistance that arise in ovarian therapy. We believe that some of these tumor cells actually undergo an adaptive response. And so essential to that process is GCN2. So if you inhibit GCN2, you might be able to kill a lot of these tumors. And then here's a cartoon I'm showing on the right-hand side of the screen, just showing you nutrient stress brought about by amino acid deprivation. But you also know of endoplasmic reticulum stress. Those are two arms that leads into stress biology within tumor cells. And so if you can actually inhibit some of these stress pathways, you can cause direct tumor ablation. Now, GCN2 belongs to a family of kinases, and they all, so I'm showing them here, they're all actually coupled through EIF2 alpha. We call them the EIF2 alpha kinases. But you could see the induction of activation of each of these kinases is brought about by certain stressing agents. So for the case of GCN2, you have amino acid deprivation, the kinase PERK, endoplasmic reticulum stress, and then you have PKR, which responds to double-stranded RNA viruses, and then heme depletion is HRI. But interestingly, all of these couple to EIF2 alpha, like I told you, and they activate EIF2 alpha to phosphorylation, and that leads to kind of a global shutdown of protein synthesis. EIF2, when it gets phosphorylated, activates ATA4 as a transcription factor, which can either drive survival in certain tumors, or you can actually drive apoptosis in others. But what's interesting from this diagram, what I'm trying to depict is of all of the EIF2 alpha kinases, a recent study have showed using DepMap cell lines that if you assess for the dependency of a broad swath of tumor cell lines on EIF2 alpha kinases by using CRISPR knockdown, it looks like GCN2 was the kinase that really drove the majority of dependence for all of the EIF2 dependent kinases in a lot of these tumor cells, which kind of boosts or give us confidence that GCN2 is really, really an essential Achilles heel in a lot of tumor types, more so than PERK. Give a lot of the integrated stress response in tumors that actually have adapted and trying to use that as a driving phenomenon. GCN2 specifically would be an Achilles heel than some of the other EIF2. And so we did this experiment, which was really, really striking, where we took OVCAR8, a representative ovarian cancer cell line, and we took SKOV3, and we treat them with L-asparaginase. Now, the idea here is to create deprivation in the microenvironment. So L-asparaginase actually synthesizes. So L-asparaginase actually depletes asparagine, whereas asparagine synthase makes asparagine. So you could imagine if you treat these cells with L-asparaginase, you're really going to deplete the amount of asparagine that's present, creating a nutrient stress. And then these cells then activate GCN2. So if you end up knocking out GCN2 out of these cell lines, you could see they become very, very sensitive. So what I'm showing here for OVCAR8, you have in blue the wild type, and I'm treating it with L-asparaginase, and you could see you get sensitivity at much higher concentration. However, if you knock out GCN2, which is the red line here, you see this huge sensitization, validating that GCN2 is one of the adaptive pathways for these tumor cells. Similarly for SKOV3, you see a very similar phenomenon. But what I want you to pay attention to is this concentration of L-asparaginase. It's about 0.1 unit per ml, and you could see that amount of asparaginase has really no effect on the viability of the cells. What I'm going to show you in a moment is that by that small concentration of asparaginase, which has no effect on cancer cell, in the presence of GCN2, you see this added sensitivity. But before I get there, I also wanted to mention to you that Rob has also demonstrated in his lab that not only nutrient stress, that's essential for survival. Some cancers try to evade, survival from nutrient stress by activating GCN2, but also when some of these cells undergo chemotoxic stress, like cisplatin, which is usually a standard of care, we realize that the GCN2 pathway gets activated. So that would be very ideal for people who are on first-line therapy, on the cisplatin treatment. You could imagine the chemotoxic stress that the tumor is exposed to. We're hoping that with activation of GCN2 under those tumors, if you come in with GCN2 as an inhibitor, these tumors would actually, undergo cell death. And so this is a finding that complements not only the nutrient deprivation, but also chemotoxic induced stress. So just to give you a flavor of the therapeutic approach that we have been using. So as I say, highly collaborative program between Rottapel's team and our group. Cascade here. So what I'm showing you here is a flow scheme that we developed for the program. So what we tend to do is we synthesize compound as a first step. We send these compounds off for a kinase screen at a company called Eurofins. Then internally, we actually looked at the EIF2 alpha phosphorylation event that's driven by GCN2 or PERG. And then after we establish some of that cellular biology and biochemical interaction, then we tend to assess some of the ADME properties for these compounds before we go into tumor models. However, at the same time, we also looked at, proliferation in vitro of cell lines. We actually have these nutrient stress-induced cell lines, where we treat them with L-asparaginase. But nicely, we have access, Rob have provided access to the UH and PDX-derived organoid cell lines. And so that gives us a lot of confidence that a lot of the lesions that you see in typical, ovarian tumors today, we saw some nice sensitivity. So I'll show you some of that data. And just generally, sometimes we want to know that our inhibitors are very specific for the kinases. So we typically do a broad kinase selectivity screen, also done at Eurofins across almost 400 kinases, just to show you that we get good selectivity. So yeah, just to introduce you to one of our early lead candidates, I'm going to call it GCN2 inhibitor OICR 36860. We stack the property of that compound up against some of the target criteria that we put in place. And I've put a lot of them here, but that is to say, our first compound, our early compound 3680 has very, very nice profile across the board. There's just a whole profile here of about 4 micromolar, which we think is kind of moderate, but that's something we've actually already demonstrated that we can improve. But it's important to note, we already have a very, very good profile for one of our early lead candidates. It's very important at GCN2 and also in cells. And then I'm just going to show you some of the property of this compound. So when we screen this compound across the entire kinome, almost 500 kinases at about 300 nanomolar, I mean, this compound have, exquisite selectivity. We hit very, very few kinases, you could see. This graph actually shows activity of kinase. So you could see GCN2 is right up here, very, very good inhibition. And so we got very, very good selectivity for a compound across the entire kinome. Now, just to show you how these compounds perform in vitro under, the condition of non-stress versus stress, amplifying that view of, therapeutic window. So here, what I'm showing you now is OVCAR-8 cells, and I choose two inhibitors here. 36860 is our internal proprietary inhibitor, and then as a tool compound is 18453, which is at least published and out in the public domain from Takeda. The nice thing about these compounds is that you could see here, they don't do much by themselves as inhibitors if you dose them. However, if you add, remember that concentration of asparaginase I mentioned to you earlier, 0.1, that doesn't do much to the cells, it's right here. And then you kind of dial in a dose response of the GCN2 inhibitor. You see this nice anti-proliferative effects with very, very good potency. And you could see our compound is actually a lot more potent than the Takeda compound. So it's a nice sensitization. So this kind of gives you a good sense that there's going to be likely therapeutic window between tumor cells that are under stress versus normal cells. But more intriguingly, when we actually test some of these compounds under very similar condition in some of the patient-derived organoids from UHN Biobank. Again, you could see the two GCN2 inhibitor by themselves on top here don't do much during dose response, but if you treat them with a small amount, 0.01 micro units of L-asparaginase in the presence of the GCN2 inhibitor, you see this nice inhibition of the growth across a number of different organoids, which got us very, very excited about this data set. More importantly, these compounds have amazing PK profile. I mean, it's unusual sometimes very so early in a medchem program that you got compound with such good PK, but right off the bat, some of our early lead compounds, 90% oral bioavailability with very, very nice clearance profile, half-lives and bioavailability. So that hasn't been a problem in the program. So we have some really strong compounds. And we took our first lead compound and just did a quick test of looking at SKOV3, just to see if you're going to get anti-tumor effects. So you can see this is just treating the animal with compound by themselves. And we see this anti-tumor effect at the level of even 3 mg per kg twice daily and 10 mg per kg once a day. So we have early hints of, efficacy in vivo. What's also nice too with this program is, again, we like to know that we have biomarker that we can check. So like I tell you, I mean, already a biomarker like asparagine synthase or ASNS, which is a good biomarker for predicting whether or not you're going to get sensitivity. We have learned, and I've just shown you some of the data, but we've had some organoids where we have looked at the ASNS status, and we realized that when ASNS status is very low, which means they cannot make much asparagine, and you treat them with asparaginase, you see this huge sensitivity telling us that if we seek for ASNS low patients, those patients actually are likely to be very highly sensitive to therapy, creating a predictive biomarker of the patients most likely to respond. And so that's always nice to have in a program. You can actually track that, and that's shown across some tumor lines here. So to sum up then, again, we have been doing this program for about almost 2 years now. And, we're in lead optimization phase. We're trying to find orally bioavailable compounds. We have demonstrated that GCN2 is a well-validated target, not only in ovarian cancer, but in other cancer type. I've just shown you some, ovarian cancer data. We believe that cisplatin treatment actually induces chemotoxic stress, which can also be used. So GCN2 can also be used as a strategy for tumors that are undergone cisplatin stress. We have identified selective inhibitors as lead candidates. They've shown very, very good oral properties. They're demonstrated good target engagement in cells and also in patient-derived organoids. We have shown proof of concept in vivo in a xenograft model, and we have actually filed a patent. So we believe that we have the potential to be best in class, and we are poised to deliver an IND-enabling candidate in the next 6 to 12 months. So we are quite excited as a group that we might have things happening in this program very soon.